Estão animados pro Jogo do Brasil? Ó, tem gente animada. Então, a gente adiantou o seminário hoje por causa do jogo, né? E hoje o CTVacinas vai ter um early dismissal, 4 horas da tarde, para a galera poder chegar em casa a tempo, torcer pro jogo. O Brasil não pode perder, hein, gente? Vai ser uma vergonha perder também para a Escócia, né? Eu tenho dúvida para quem que o Simon tá torcendo, porque ele é do Reino Unido, né? Não sei. Mas ele tá aqui, então... Se ele ficar... Yeah. We are wondering if you are cheering for Scotland or Brazil today. Brazil, of course. Claro. Bom, o Simon já interagiu com vários de vocês, mas é um prazer receber o Simon aqui. Ele é formado em bioquímica com doutorado em imunologia, né? E desde o doutorado dele, ele vem trabalhando com vacinas para malária, né? E atualmente ele é professor em Oxford e é professor de vacinologia e medicina translacional e também é vice-chefe de departamento, né? Com o George, que também já esteve aqui. E ele foi apontado como, né, foi eleito para a Academia de Ciências Médicas da Inglaterra, que também é um big deal, né? Então, pesquisador jovem com bastante sucesso já. O trabalho dele é muito focado em vacina para malária, especialmente, né, anticorpos, né? E ele vai falar um pouco disso para a gente. E na pesquisa dele, eles desenvolvem as vacinas, fazem os testes pré-clínicos e levam para o ensaio clínico. Então, ele tem vários ensaios clínicos, né, de fase 1 e 2, realizados especialmente na África para vacina de Plasmodium falciparum. E está desenvolvendo um modelo de infecção humana controlada para Plasmodium vivax. E foi aí que a gente começou a interagir, né? Eu estava numa reunião que eu falei, a gente tem uma vacina para malária que a gente está desenvolvendo, para malária vivax, e o consórcio que ele lidera para desenvolver uma vacina para malária vivax, né, e que está financiando esse projeto de desafio humano para malária vivax, estava precisando de uma vacina para testar. E a nossa foi assim, encaixou perfeitamente com a necessidade dele. Agora a gente só precisa da Anvisa deixar a gente andar, né? Então a gente continua. So, Simon, I just told them your CV in Portuguese, which you know already, so you are fine with it. Yeah. But thank you so much for taking your time and coming to Brazil. I hope you enjoy. And now the floor is yours.
Obrigado, Helton. Afraid that's the only word of Portuguese I know. So.
She wants to give a...
Are we good?
Yeah, we're good.
All right. Yeah, sorry, I cannot do this in Portuguese. But thank you very much for the invitation and your warm hospitality. It is a real pleasure to be here and a real honor to be in Brazil for the first time. I'm really enjoying my visit. So I think Helton told you that I obviously work on malaria. What I thought I would do today is tell you two stories. So in the first half of the talk, I will tell you about some of the recent work we have done on vaccines for falciparum malaria. And in the second half of the talk, I will switch to Plasmodium vivax and tell you about work we've done with the human challenge model, which I hope you will find of interest. So just, I think as Helton told you, our lab is based in central Oxford. This is the team here in the photo. Helton and Greg have both visited us. If you're ever in Oxford, you are most welcome. Let me know. But the work, the vaccine development and the immunology is done in a new building in central Oxford. And I work in partnership with Professor Angela Minassian. She's the real doctor, and she runs our clinical trials program, which is very busy and very active across trials in the UK and Africa. And that work is all led by her. So I suspect you're familiar with malaria. I know you have a program here, but just a reminder, the parasite has a very complex life cycle. You catch malaria from the bite of an infected mosquito. It puts sporozoites into your skin. They look like little eyelashes swimming around down the microscope. They go rapidly to your liver, where for falciparum malaria, they are there for about 7 days. They then rupture out into the blood, where they undergo a continual cycle of growth in the blood. And that's when you get the symptoms and the associated morbidity and mortality. Some of those cells some of those parasites turn into sexual cells called gametocytes. They are taken back up in a blood meal of a new mosquito. They fuse in the midgut to form a cyst, and over 2 to 3 weeks develop back into sporozoites. So I'll come back to this throughout the talk, but it's a really, really complex biological process, and so very hard to stop by vaccination. And I will add that there are in fact 6 species of Plasmodium that infect humans. In this first part of the talk, as I mentioned, I will focus on falciparum. So as most of you will know, malaria is endemic throughout the tropics. In the case of falciparum, over 200 million clinical cases every year, leading to just over 600,000 deaths, primarily in young children in sub-Saharan Africa. It's not all doom and gloom. There is a lot we can do with malaria control, including drugs, insecticide spraying to kill mosquitoes, and sleeping under bed nets. But that being said, this graph here shows you the number of deaths globally over the last 30 years. And you can see that despite malaria control, despite $2 to $3 billion a year being spent on bed nets and drugs and insecticides, we still have over 600,000 deaths per year. And so we need a transformative tool. And that is the real hope here, that a vaccine could be such a tool to help drive down those number of deaths and bring malaria further under control. So many of you will know malaria vaccines have been a real challenge. I've already told you that we have this highly complex multi-stage life cycle. The parasite has lots of immune evasion strategies, and we have huge antigenic diversity to contend with. And so those are the main reasons malaria vaccines have been so difficult. But that being said, researchers are finding chinks in the parasite's armor, and I'll come on to talk about some of those. But even when you find those, you still need really strong immune responses for those vaccines to work. So one of those chinks in the armor is a protein called CSP, found on the surface of the sporozoite. We've known about it for many decades. You can see here it lit up in green. This protein has globular domains at the N and the C termini, and throughout the middle are runs of amino acid repeats. In the case of falciparum, they are 4 amino acids long. And we've known for many years that if you can get high levels of antibody against those repeats, that those can be protective. That's been shown in preclinical models and more recently through human clinical studies where they have passively transferred CSP biologics, and they are protective. So how was this taken forward as a vaccine? Well, some of you will know that we now have 2 approved vaccines. one called RTSS from GSK, and another called R21, developed by my colleague Adrian Hill in Oxford, and now produced by the Serum Institute of India. Both of them are very similar. They are the hepatitis B vaccine, so the surface antigen of hepatitis B, and on the surface is arrayed the CSP protein. So the main difference between the two vaccines is R21 has a higher density on the surface of CSP. They're given in really strong adjuvants with saponin-containing formulations, AS01 from GSK or Matrix M from Novavax. 4 doses are given to 5 to 17-month-old children. So 3 in the primary series at 5, 6, 7 months of age, and then a booster in the second year of life. And so you get these very high antibody responses that, of course, wane over time. These are the real data. You can see them coming up and then declining. And then here you have the boost in year 2, and they go back up and then decline over time. So those went through phase 3 trials, both vaccines. Depending on the study, they are 50 to 75% effective in the first year at preventing clinical malaria in young children. They have a good safety profile, they're cost effective, they're approved by the WHO, and they're a huge milestone for malaria, the first approved vaccines, but also a huge milestone for human vaccinology. They are the first vaccines against parasites that are approved for use in humans. So that brings us to the question about where next? Where do we go from here? So there's been a lot of discussion about this in the field. Of course, we can still look for better efficacy. The WHO target is for 90% in children. With the current vaccines, immunity wanes over time because the antibodies come down. And in the real world, there is very low uptake of the 4th dose in the second year of life. So there's room for improvement. And of course, when one parasite, just one, gets through the liver and into the blood, then the individual is at risk of malaria disease. So one of the main approaches the field is looking at the moment is to add in more components around the life cycle to give you multiple lines of defense in what we call a multi-stage approach. You can think about this in different ways. For children, where the risk is highest of death and disease, adding in a blood stage component would perhaps be a sensible first step. But there's a lot of discussion in the field as well about now older children and adults, because although they are immune or naturally immune, they are transmitting, they are the reservoir. So for elimination, we probably want to stop infection or transmission in adults. So there's work going on with what we call transmission blocking vaccines that work in the mosquito. So I don't have time to talk about those today, and my own group has worked on blood stage vaccines for 2 decades now. So I thought for this part of the talk, I will tell you about that. So this is the blood stage parasite. I told you it has this exponential cycle of growth every 2 days. This short video here shows you what we're up against in terms of the biology. Here is a yellow merozoite coming in. It attaches to the cell, reorientates onto its nose, forms a tight junction, and pulls itself in to the red blood cell, shedding its surface. So just to reinforce, really complex biology. Loads of protein interactions happen during invasion. There's a lot of variation or polymorphism. There's redundancy in the system, so spare keys to the lock. And those have been real challenges for the vaccines. And for many years, a lot of really good groups tried to make a blood stage vaccine, looking at the surface proteins, MSPs, AMA1, which forms a tight junction, erythrocyte binding antigens, and sadly, in phase 2 trials, none of them worked. So about 15 years ago, the field was in a real miserable, unhappy place. And then in 2011, we identified a protein called RH5. And this has really changed the field over the last 10 years. So my group stumbled across RH5 by reverse vaccinology, so antigen screening. And at the same time, Gavin Wright's lab in Cambridge in the UK identified its receptor Basigen, and that they showed this interaction is essential. There's no spare key, there's no backup. And if you block that interaction with antibodies, if RH5 doesn't meet Basigen, it's game over. And everyone said, well, that's too good to be true. But 15 years later, nobody has found an exception to the rule. RH5 is also really conserved. There's hardly any polymorphism. So it was a big surprise and has become one leading strategy to develop a vaccine. So that was the discovery. Matt Higgins' lab in Oxford got the structure in 2014, so we could visualize it for the first time. RH5 has this diamond shape in yellow, so 2 bunches of alpha helices that dock into Basigen on the surface of the membrane, which has 2 immunoglobulin domains. Our first in vivo data came from looking in a non-human primate model. I was saying to Helton, we don't have many collaborations in South America, hopefully more to come. But this was our only one with the team, the US Navy in Lima in Peru, where they have a non-human primate facility. And Aotus monkeys are the only species you can infect with falciparum malaria. So here you see control animals. This is thyme with parasites in the blood. And you can see falciparum grows very rapidly in the blood, and you have to drug treat the animals. Here were the animals vaccinated with RH5, either no parasites or control and clearance. So that was the best result in this model at the time, and we were very excited to get those data, probably back just over 10 years ago now. We tested different vaccines in the model, and we saw a spread of protection, so we could ask if there was a correlate. And working with Carol Long at the NIH, they have an assay we call GIA, or growth inhibition activity, very similar to a virus neutralization assay. You culture the parasite, you add antibody, and you ask how much growth is inhibited. And we saw in non-human primates, here is the GIA in vitro, here is the in vivo protection. We saw this really strong correlation between the assay and protection. And if you could get the animals above the green line, over a level, they were protected. So that gave us a target to aim for in humans. So we wanted to translate this to the clinic. RH5 was really hard to make. No bacteria, not in yeast, barely in mammalian cells. It was a complete pain, not just for us, but everybody. We eventually made it in Drosophila cells, S2 cells, with a Danish company. You can see the blob here on the gel, 60 kilodalton RH5. Then it was a nightmare to purify, but we used a new technology called CTAG, which is 4 amino acids at the C terminus. That was with a Dutch company, but it's now commercialized by Thermo Fisher, so you can buy the resin. But we took these through GMP for the first time. It was approved by the UK regulator, these new technologies, and we made a batch of protein to GMP and then for clinical trials. This was done at our facility in Oxford. It's since been licensed to Serum Institute of India to upscale. And since then, we've taken RH5.1, this protein, with matrix M adjuvant, through a series of clinical trials in the UK, phase 1 in Africa, in Tanzania, and then phase 2 in Burkina Faso. And I'll just show you a couple of highlights from those studies. So essentially, over those phase 1 trials, we optimized the dose of the vaccine and the regimen and how we delivered it. And this is the GIA in humans. So we started off here, and over 10 years, you can see we iteratively improved. And the red line is the target from aotus monkeys, from the monkey model. We were trying to get over the red line. So we started here, but finally, in our phase 1 in Tanzania, we got up here and all the children were over the red line. So we were very excited. That took us 10 years to get from here to there. But then we could ask, now we've hit our immunogenicity target, does the vaccine protect young children from clinical malaria? So we went on to do a phase 2b trial, working with Halidou Tinto in Burkina Faso. He has a fantastic, fantastic team who do trials there. This was a double-blind, randomized controlled trial in 360 healthy 5 to 17-month-old children. We had 3 groups in the trial. Controls received the rabies vaccine, and then we gave the Rh5 vaccine in 2 regimens at 0, 1, and 2 months, or 0, 1, and 6 months, a delayed boost. So in terms of immunology, the vaccine was highly immunogenic in the vaccine groups. There were no responses in the rabies control. And this is the GIA again, here with the 2 vaccinated groups, over 80%, close to 90% of children over the target, and none in the controls. So we were very happy with the immunogenicity. We then went on to monitor their children for clinical malaria. So they were monitored for 6 months over the malaria season. The primary endpoint was time to first episode of clinical malaria, defined as fever plus parasites of over 5,000 per microliter. Based on the history of this type of vaccine, we thought we were braced for 0 efficacy, but we were very happy to see with the delayed 3rd dose, 55% efficacy, which was significant on the primary endpoint. We also then looked at different definitions of malaria, and that shows you here. So 5,000 is the one we normally use. It was slightly lower for any malaria, over 44% efficacy, but Roth regimens showed 80 to 90% efficacy against high parasitemia in the young children, suggesting that you were pulling down parasitemia. There was basically no high parasitemia in these young children. And we think that's important because although we don't have the data yet, we think that would mean it would prevent severe disease. And that is what we've seen for 2-3 decades of animal models, whether it's aotus monkeys, rhesus macaques with Knowles eye malaria, mice with Plasmodium yoelli. All of the controls show high parasitemia. And then with prototype vaccines, you see control, some no parasites or others control and clear. You're pulling down the parasitemia. And that's what I think we're seeing in our phase 2. So that's been really exciting. It's the first blood stage vaccine that does anything in a phase 2 trial. So other work that we do at the lab looks at the human antibody response. We do this to inform vaccine design, to compare human vaccination strategies, or even to identify potent antibodies that we could use as biologics. So as a lab, we have spent many years setting up an infrastructure to isolate human antibodies from the clinical trials that we work on. So this is always evolving, but the current pipeline involves isolation of antigen-specific B cells. We now do 10x to isolate, 10x single-cell genomics to isolate the antibody coding genes. We then do high-throughput cloning and antibody production, and then high-throughput characterization by ELISA, SPR, and functional killing. And we've looked at now well over 1,000 human antibodies to Rh5. And just to give you a flavor of those work, we published a lot of the data a couple of years ago. We pulled out lots of antibodies from people in Oxford that we have vaccinated. We have a high-throughput SPR machine called a Cartera, which enables you to look how the antibodies compete for binding. And you get these network plots from the Cartera. So these colored blobs are groups of antibodies that compete for binding. So they're sort of regions of the surface of Rh5. And then we can start to analyze what each of these groups does in terms of function and potency and characteristics. And to cut a long story short, there are 5 main antibodies to Rh5, types of antibody. So here in the middle is, in gray, is Rh5. The receptor binds at the top. And these antibodies in blue, purple, and red are essentially the killing antibodies, the inhibitory ones. We have other antibodies in green that bind down the side that don't do much, and also antibodies in yellow. Sorry, my team should be off. I thought I'd turn that off. Other antibodies in yellow that don't kill the parasite. So this information is now being used for sort of 2nd generation and 3rd generation designs by other labs that we're supporting, who are doing a lot of immunogen design to focus on specific epitopes. And then finally, just for this part of the story, I told you earlier we have this interaction between Rh5 and pathogen. Well, the biology has got more complicated in recent years. A lot of work from Alan Cowman's lab in Australia has now shown that Rh5 is just one component of what is in fact a pentameric complex. There are 5 proteins, all conserved, all essential, and all interesting vaccine targets. And they dock into complexes on the red blood cell surface. So we've been studying these other targets, especially this green protein here. It's massive. It's called Ripper and very hard to work on. But we published some data just last week, a partnership between our lab, Barney Williams, a great postdoc, and Jordan Barrett at Scripps Research in the US in Andrew Ward's lab, and where finally we got the structure of Ripper for the first time. So it has this body here, then this long tail that sort of snakes up and docks onto the membrane. And no one was able to get the structure of this until Jordan attached 6 different antibodies. So here you see the body, and all of these are different antibodies bound to that tail. And what we've shown in this paper is that as those antibodies bind along the tail, they restrain its conformation so it can't move around anymore. And that's how they are inhibiting parasite growth. So lots of exciting stories coming out from around that complex. We since made another vaccine called R78C based on that tail region and this blue protein here called CYRPA. And we've combined that with RH5.1, the vaccine I was telling you about earlier. And we took that into a phase 1 trial in Oxford as well. We made this in the same way using the Drosophila S2 cell platform. So these are the human GIA data, the functional killing. In blue is Rh5. This is 2 different antibody concentrations. We were really happy to see the new vaccine in red also gives GIA in humans, but the combination of the 2 in purple is the highest GIA we've seen to date in human vaccinees. So that is what we're taking forward. So the goal now is to combine this new blood stage vaccine, which is RH5.1 and R78C, 2 proteins, and we're going to add those to the R21 matrix M vaccine, which is already approved, because we think that's the fastest route for us to get to licensure in due course. And that project has just been funded by EDCTP. So this is a European Commission funder to support trials in Africa. It's led by Angela in Oxford and Magloire Natama in Burkina Faso. We have 10 partners across 7 African countries. It's a 5-year program to take the new vaccine from phase 1 and then into phase 2, combining the approved vaccine with the new blood stage vaccine. And we'll cross our fingers and see how we get on. So that's where we are with falciparum. In the last 10 or 15 minutes, I am going to completely change and talk about Plasmodium vivax, which I suspect many of you are familiar with because it's also endemic in Brazil, especially in the Amazon region. As a parasite, vivax has some different challenges to falciparum. It forms an extra stage in the liver where it goes dormant, called a hypnozoite. So you can get, although you get a primary infection, you can get relapsing infection over many weeks, months, or even years. In the blood, it is tropic to young red blood cells or reticulocytes. And it also transmits faster. So it often transmits before people know they are infected. One huge problem with vivax is that you can't culture it in the lab. So assay development and studying the biology has been really hard because you can't culture it to study it. So as I just mentioned, vivax is endemic in South America and across large parts of Southeast Asia and India and the Horn of Africa, but not sub-Saharan Africa or Central Africa. We've known why for over 50 years now. Lou Miller reported in 1976 that this was associated with the presence or absence of the Duffy blood group. So to invade a red blood cell in the case of vivax or a reticulocyte, you need an interaction between the Duffy binding protein on the parasite and the Duffy blood group antigen on our red blood cells. And in African population, their red blood cells have become Duffy negative, so the parasite can't invade. We have been lucky. It's hard to get funding for vivax, but we've been lucky over the last 10 years to have funding from the European Commission, despite Brexit, which is one of the worst things my country has ever done. But we have been lucky to have European Commission funding. We had a 5-year project running from 2017 to 2022 called Multivivax with partners from across Europe. And in that project, we focused on the blood stage of the parasite at the time to build the tools that we needed to work on it. So we wanted to set up a human challenge model. We developed new functional assays, this work led by Rob Moon at the London School, where he was able to adapt Plasmodium knowlesi, which is a rhesus macaque malaria, to in vitro, and he can change their genes to the vivax ortholog, so you have transgenic parasites for assays. And we wanted to test the vaccines that were available or people had made to Duffy binding protein. So I'm going to tell you about the work we did to set up this part of the human challenge model. So you may be familiar with the fact that the human challenge model for malaria has been around for many decades for falciparum, and there are different ways we are able to infect people or consenting volunteers with malaria. So traditionally, they have received 5 mosquito bites. So in our case, we take volunteers to London, to the insectary. They put their arm on the Starbucks coffee cup, and they get bitten by the mosquitoes. 5 bites. There are frozen sporozoites for falciparum, which you can get from a company called Malaria in the US. And when we do that, we then monitor the volunteers by PCR. They come to the clinic every morning and every evening. We take a blood sample and we do PCR. And here you see on day 7, 6.5 or 7, out they come from the liver and they start to grow in the blood every 24 hours. And at about 10,000 parasites per mil, the volunteers are drug treated So just to assure you, everybody survives, everybody gets the drugs, everybody has a good experience. But this is basically just as they're getting symptoms. So some volunteers have no symptoms at all, probably 50%. The other 50% starting to get symptoms. From the data, we can look at what we call the growth rate, how fast they're growing, or how many come out of the liver and into the blood. The other model is blood stage, where we inject blood with parasites in directly intravenously. And here, this is, again, Oxford adults. You can see we monitor them and you can see them growing. It's really reproducible. Every 24 hours, they grow tenfold and they're drug treated at the same time, and we can get the growth rate in the blood. And of course, if you've given a vaccine or a drug, you hope to see no parasites or perhaps reduced parasites, depending on what your question is. So we wanted to set this up for Plasmodium vivax. To do this, in order to develop the model, we needed to get blood from a donor which was infected with vivax. The question is, how do you do that? Where do we get the parasites from? How do we find volunteers infected with vivax to donate the blood? And we had to, as a group, establish how we could freeze down this blood to produce a bank of parasites that was safe and suitable for human challenge studies. So the first question was, how did we get them? Well, one option was to wait for a person to come to a hospital in the UK with vivax. And this is meant to be Angela at the bottom. We thought about this for a long time. We do get vivax, but we don't know when. We get returning travelers or perhaps family members who catch malaria vivax when they're abroad and come back to the UK. But we don't know when they're going to come to hospital. We don't know where in the UK. It could be the middle of the night, could be any time of the year, and we have to be on standby to somehow take their blood before they get treated. So basically, that was impossible. But they do that in Australia. So the Australian group are doing that, but impossible. So we thought it was much better to infect volunteers by mosquito bite at a time of our choosing to infect them. The question is then, how do we get the mosquitoes? And we turn to partners in Thailand, Sajetsaman Prachumsri, who has a lab in Bangkok. And she has great sites in the north and the south of Thailand where she can get the blood to infect the mosquitoes. So that's what we did. She went to her sites. She took the blood back to Bangkok and fed mosquitoes. The blood was 10 days to do the safety screen. She infected the mosquitoes and they were flown 2 weeks later to London, where we had 2 people ready and waiting, once we knew the safety was okay, to infect them by mosquito bite. And these 2 volunteers we had health screened. We knew they were universal blood donors. They were basically as healthy could be to produce the bank. And then, assuming the parasites came into the blood, they would donate half a unit of blood and we would freeze it down in the lab. So this is what happened. Here we are on day 7. You can see in both volunteers, out the parasites came, it grew. And then at this point, they each donated blood and we froze that down under very controlled conditions in the lab. I actually haven't got a photo of that. We wanted to do it in the health service, but they would not touch malaria blood in the UK hospital. So we had to do it ourselves and establish a clean room in our lab to freeze the blood. So it was a lot of work, a lot of stressful fun, but a lot of work. So it was GMP-like. We tested for sterility, mycoplasma, endotoxin, all okay. The volunteers had more health screens, all okay. We could see parasites in our blood by microscopy, which was good. We didn't kill them during the freezing. We sequenced the genome, which was the highest resolution genome reported at the time for Vivax. And we had 190 vials of blood suitable for human challenge. So we were really pleased with how it went, but a huge team effort. We then took that blood once we had finished all the analysis and the paperwork and got approval for a new trial to infect volunteers to check it worked. So we infected 6 people here on day 0, and we waited. And then here come the parasites growing in the blood on day 6. They come up on day 6, 8, and you can see them growing. So it worked in all 6 people, and we also got sexual gametocytes. So we were very happy with the blood bank. We then went on to test some vaccines against Duffy binding protein. One was an adenovirus and MVA, pox virus that we'd made in Oxford. Another was a protein. I won't dwell on the vaccine testing. One of the candidates actually inhibited the growth, so it was very exciting, and we published those data a couple of years ago. What I want to finish by is telling you what we've done most recently, which was our new consortium, funded in 2023 for 5 years, again by the European Commission. We were very grateful, despite Brexit, that we were able to carry on with the project through UK government and Swiss government funding. So we've got a new team of partners across Europe and one from Ethiopia. It's a really fun project and fantastic group. Again, trying to improve the tools that we have for Vivax. So we're making new types of transgenic parasite, new types of assays, and trying to do more with the challenge model. And I'll finish just by showing you work we've done on the challenge model. So here, we've done the blood stage. We now wanted to do the rest of the life cycle, because that hadn't been done before. So we wanted to assess whether it was safe and feasible to look at relapsing from Vivax in a human challenge model. Importantly, using a defined clone, so the clone we just made from Thailand. And this has hardly been done before, but normally people go and get mosquitoes from the field, and you don't know what the strain is. It might be a mixed infection. It's really hard to do. But our plan was to infect people by mosquito bite, wait for them to develop their first infection, and then treat them with the usual drugs, but not primaquine, which is used to kill the hypnozoites in the liver. And we would then leave them for a 6-month period to see if they relapsed, and then give them primaquine after 6 months. And again, this hasn't been done before in a human challenge model. We took a lot of thought about how we were going to do it safely, because people can't come to the clinic every day for 6 months. That's too much. There was only one problem, though, in that we needed the mosquitoes. How were we going to get the mosquitoes again? So here we have a fantastic partnership with the group in the Netherlands at Radboud University. They're really fantastic at mosquito work. So they were able to take our Vivax blood that I just told you about. They infected volunteers in the Netherlands and fed their blood to mosquitoes, and were able to produce a batch of mosquitoes. In real time, Oxford recruited new volunteers, and because we couldn't go to Imperial College because they closed their insectary, we actually flew our volunteers from Oxford to the Netherlands. So this was an international CHIM model. So there's hope, Helton. There's hope. We flew them to the Netherlands, where they received their mosquito bites, and then we flew them back to Oxford to monitor the infection. So this was huge logistics between 2 clinical teams. And I should say both studies went on medRxiv yesterday, so the data are now out there. Yes, and on 15th of April last year, they got challenged. So here in the Netherlands, they had their 5 infectious bites. You can see they don't use Starbucks coffee cups. They have these great little boxes. You can put the mosquitoes in. The volunteers were bitten. And then we monitored them from day 7. And all 5 of them got malaria. You can see they came out of the liver, and we drug treated all 5 within 12 days. So that was their first primary infection. All developed parasitemia and all cleared. And then the million-dollar question was, well, what happens over the next 6 months? Will they relapse? And this was a really fun study to do. They came to the clinic every 14 days, unless they felt sick, in which case the doctor was on call 24/7 and the lab. But in short, over the next 6 months, all volunteers relapsed at least once, and 2 volunteers relapsed twice. So you can see here the different colors of the different people, but you can see the relapses happening over that time period. It was a lot of fun to do, and really rewarding, I think for the lab.
So you didn't use
Not till the end.
until the end.
Only at the end. Yeah.
But did you treat them?
They got treated each time there was a relapse. Yeah, they got treated, which is why it goes away. So sorry, yeah, so here, this would be detected, then treated and gone. And the green one never came back. The red one came up here, treated, and then another one here, treated. So in summary, this is the first completion of the whole life cycle with a defined clone by CHMI. We hope the model can now be used for vaccines, drugs, diagnostics, or experimental medicine. We have the protocols established for each step of the life cycle, and very happy to share those with other partners. And in terms of our own project, we now plan to go on and test a CSP-based candidate vaccine with the new challenge model. And I should add, of course, that our original clone was from Thailand. We would love to do this again, but with a clone from another continent, perhaps this one here. So I will just finish by acknowledging huge numbers of people. This is the team who work on the falciparum trials in Africa. We have amazing partnerships with many teams without whom the work wouldn't be possible. Our own lab in Oxford, as I said, all our partners, too many to mention here, the funders, and you for your attention, and happy to take any questions. Thank you.
Thank you. Thank you, Simon. That was fantastic. Like, I wish we could do that in Brazil, but I think we are.
You will get there.
Yeah, hopefully. So, any questions?
Hi.
Hi.
My name is Beatriz. Thank you very much for the presentation. It was really, really great. I have actually 2 questions about the model for Vivax. The first one is about the symptoms. So when the patients, they get the relapses, they usually get symptoms. They get some symptom or not. You just identify the infection because of the constant monitoring, or if they get symptoms. And the second thing is about the cryptic niches that we know that we have in Vivax. So we don't know how long do you need the patient to have a parasitemia to form this cryptic. I'm so sorry, this word is so
cryptic. Don't worry.
Yes,
Yeah,
I'm so sorry. But we don't know how long it needs, or if multiple infections are needed to form this. So do you guys evaluate it or something like that?
When you say cryptic niche, you mean the mass in the spleen?
In the bone marrow,
in the bone marrow.
Yeah.
Yeah, okay. So to answer your first question, we discussed how often they should come to clinic. We settled on every 14 days. We were going to do every week, but we ended up on 14 days. Most a lot of them were caught, well, there were only 7 relapses. I think 5 were caught by coming every 14 days, but there was one period, I think it was those 2 there, that were symptomatic, so they came, or they had a headache, so they came in, and they were PCR positive. But we thought 14 days was probably the longest we could go, before they got bad symptoms.
Because there's a really high parasitemia.
So. Oh, I know. Yeah. So some of them were really high.
Yeah,
but the symptoms were no worse than we've seen. The symptoms, okay. The data's in the paper, but that in the clinic they are really high. You're right. So some of them had obviously grown up before we caught them.
Yeah.
But they are higher than we would normally see in a primary infection, where we treat at 10,000.
Yeah, my question is not about, I'm sorry, and maybe be a little bit rude what I'm going to say, but it's not about like actually the safety. I am interested about the biology basically, because if the patient didn't have any symptoms, and we do see that a lot here in Brazil, we had asymptomatic individuals, the parasitemia changed a lot, so it was more about the biology.
Yeah, well, hopefully, I mean, obviously, in human challenge studies, safety comes first. That is always the most paramount. I mean, these are naive UK adults. In an endemic area, people will have perhaps had many exposures and acquired some immunity. I mean, the amazing thing is now, is all the amazing experimental medicine things people are doing, sampling lymph nodes, fine needle aspirate, taking bone marrow samples. We've just done in another trial, aspirate and biopsy, so you can actually look in the bone marrow. I haven't seen anyone do spleen yet, but you can get to the bone marrow now. We've been doing labeling cells with heavy water. There's really cool stuff being done by the community. And perhaps, hopefully, when you can do challenge studies here, you may be able to do this in an exposed population and do similar studies. I agree, they're really cool questions. I don't have an answer for you. Thank you.
Well, thanks, Simon. Amazing work. And just to get the car that she brought, I didn't get. Most of these individuals were not symptomatic.
Not bad. Not worse than normal. I'd have to look back. So I don't know off the top of my head which ones.
They're mostly
they were mild they were mild symptoms. Yeah.
I see. I was wondering what is the natural immune response against the RH5 and the Riper in South Africa. Those the individuals when they get older, they have more antibodies.
No. So that the RH5 complex is really interesting because even in African adults, there's hardly any antibodies. Most are negative. So we don't know why you get falciparum over and over and over again. So MSP1, AMA1 always come up. The RH5 complex. People find responses, but they're really weak. And a lot of people, they're absent, which probably explains why it's conserved. But I don't know why. We're asked that a lot.
That might be some sort of immune escape from the parasite.
Quite possibly, but not sufficient to avoid vaccine-induced antibodies.
And is it also expressed in sporozoites?
Good question. Blood? So RH5 CYP Ripper, no, but it's just been reported by the Australian group that the 2 at the top, I didn't mention them, CSS and PTRAMP, they're very new, are also on the sexual stages and possibly the sporozoite. So everybody is rushing now to look at whether they work on the other stages. I think they've just had a preprint on that.
Great, thank you.
Yeah.
Well, is there any more questions? So this RH5, have you compared it to like human genome or some sort?
Good question. We did, but way a long time ago. There's no homology to human proteins.
Yeah, because
we worried it might be tolerance in the beginning.
Yeah.
But there've been no problems with vaccination in mouse, rat, rabbit, monkey, human. So there's no overall tolerance. I don't think that's why we don't respond to it in natural infection, but it's a good point.
Yeah, okay. Simon, thank you so much.
Pleasure.
For being here, for taking your time and sharing this amazing work with us. And hopefully we can fix the lack of South America collaboration.
I hope so too.
Yeah. Thank you.
Thank you very much.